Hi, this is
Hello.
hello. Uh, this is Science Hangout from Science Mission. We have Dr. Chris Colwell with us from University of California, Los Angeles. He is the full professor of Psychiatry and Behavioral Sciences at UCLA. Dr. Colwell's lab has focused on understanding the mechanisms underlying circadian rhythms in mammals. Dysfunction in the timing of these daily cycles is a key symptom in a number of neurological and psychiatric disorders. Dr. Colwell has won Outstanding Graduate Student Teaching Award in 2012-13. Welcome, Dr. Colwell.
Thank you. Glad to be here.
So thanks for joining us. Can you please tell us about your background, your education, your interest in few words?
Sure. Yeah, just briefly, I started working in science in high school, where I interned in the Food and Drug Administration. And then I attended Vanderbilt University in Nashville, Tennessee, where I became interested. I didn't know exactly what I wanted to do there, but I worked in the science library for my first year, shelving books, because people actually read books in those days, and we would have to, someone had to shelve them, and that was my job. And I kept as I would shelf things, I would just sort of read through them, and I kept being drawn to sort of the section of the library that was focused on the neural controlled behavior. And so that really was I was just 18, but I really became interested in that, and started working then in the laboratory of Terry Page at Vanderbilt University in the biology department. His research focused on circadian rhythms. I wasn't, at that point, I was really just interested in trying to have a system where I could have a chance of understanding how the brain controls behavior. And there's something very appealing about rhythmic repetitive behaviors, because they keep going on again and again. And so, you have a chance to study the mechanisms in a way that's easier than some other processes because of their rhythmic nature. So I started working then, and I was 18 in Terry Page's lab. And I went on to graduate school with Mike Menaker, who's a leader in the field of circadian rhythms in mammals. So I worked with him first at University of Oregon, and then he moved to University of Virginia, where I ended up getting my PhD. I worked with him, and then also with Gene Block, another scientist who was at University of Virginia. Then, although now sort of in a weird turn of events, he's actually the chancellor here at UCLA. So he's actually my boss's boss here, and runs the university. But at that point, he was just he was a scientist there at in Virginia, and I basically was studying circadian rhythms throughout this period of time, and became interested in understanding then their role in disease processes, because basically every patient that comes into the psychiatric hospital here, or the neurological clinic, is having some sort of disturbances in sleep. And so I started then working in a postdoc here at UCLA, in a lab that focused on movement disorders like Huntington's disease. And even though you don't think anything about something like Huntington's disease, why would that have anything to do with sleep, right? But it turns out the Huntington's patients, just like the Parkinson's and the Alzheimer's patients, the first or one of the first symptoms that the patients actually have is disruptions in their sleep-wake cycle. It occurs well before anything else. So before the onset of the motor symptoms, before the onset of the cognitive dysfunction, is this disturbances in sleep. And this is really something which is driven sort of the work that I'm interested in and our lab is doing, is trying to understand the interrelationships then between the circadian timing system and the disease process.
Okay. And eventually, you got into academics. So were you thinking about moving to the industry, or what was the thought there?
Yeah. Okay. I was always interested in sort of in academics and or medicine. That was, to me, that was where I was debating, and so being sort of in a biomedical research institute is a good fit for me. I did have a period of time when I was a postdoc, like many postdocs, I was struggling to get a job, and I was trying to figure out what the heck I was going to do. It was the funding was, the funding comes and goes as these things. Right now we're in a down cycle, but it was also true when I first was looking for employment, and so I did have some interviews with companies. In fact, the best job offer that I got was from a small biotech company in Boston, and it was really like, in many ways, it was sort of it had all the aspects that I wanted, in terms of, they were generous setup package and, supposedly a lot of freedom. But my concern about it, and why I didn't end up taking that job, was the sort of trade-off when I talked to my friends who were in especially the pharmaceutical industry. They were at that point, and I think it's still true, telling me stories about how they would be working, say, on cognitive processes, drugs that enhance cognition, and depending on how their drug comes and goes, one of my friends was in a situation where then he was told, well, either you're going to be fired, or you're going to have to switch to pain, and and so now for some people, some of my friends, that worked out great because they were really driven by the technique, and they didn't care whether they're working on cognition or pain. It didn't matter to them, right? Mm-hmm. But for me, it did matter. I really have been really focused on both the neural controlled behavior, but then circadian rhythms and sleep. So I decided in the end that my inflexibility made me better suited for for academics, and okay. And I guess most of my many of the people that work with me would agree that sometimes I can be bullheaded, and I think this was a good situation. So yeah, I think for industry, you have to be more flexible than I guess I was at that time.
Okay. And like in your career, there are several people who would have helped you as mentor, mm-hmm. coaches. So how did it work out for you?
Yeah. Well, certainly I trained with a group of really outstanding scientists, starting really at Vanderbilt. Mm-hmm. Terry Page, here this 18-year-old came into his office, and I had the audacity. I said, well, I didn't want to just work in his lab, is that I wanted to do electrophysiology in his lab. And, so I was like very specific, and most of the people I talked to, the faculty at Vanderbilt were like, they patted me on the back and said, come back in a few years. but Terry, to his credit, took me seriously, and he basically had set up some equipment and had a book of like introductory neurobiology laboratory exercises, and he basically gave me the equipment and some empty lab space in this book and said, go to it, chum, And so I spent the first summer working alone. He was off in Japan, so he wasn't even there, okay. But going through this lab exercise, and so I've sort of so certainly that was very important for me. It got me started off, and then later on, my PhD advisor Mike Menaker and my first postdoctoral advisor Gene Block were both also incredibly important. Here at UCLA, I was had the luck to work with a scientist named Mike Levine, who works in motor control and Huntington's disease. Okay. That also, obviously, he was an important mentor for me, especially on sort of practical things about getting a job and trying to survive within the UC system. He's been very helpful. So I guess I've had a number of mentors that have been helpful to me through the years.
And being in the sleep field, circadian rhythms, what motivates you not to go to sleep?
Yeah, that's right. I have to be, I have to be a little bit fair, right, to people in the lab. You can't. It's not good. All of our data says it's not good to work through the night. So. No, I actually, one of the things that and I guess many people are like this. I got into being interested in circadian rhythms, and sleep in large part because I was such a terrible sleeper. You know, it was so. And that wasn't the main reason, but it was one of these things that I have gotten better about as I've gotten older and more rigid in my schedule than when I was young. I like to stay up late into the night, and if I was excited about an idea, I would just stay up working, and and that was fine. But the reality is that that's something you can do as a young person, and but once I started having a family and stuff like that, it became impossible to live that kind of life anymore.
Yeah.
But the work that we do does have a number of practical implications for both for all of us in terms of getting a better night's sleep.
Yeah. And can you please tell me about your lab and your postdoc grad students, a little bit more on that?
Okay, sure, yeah. So in general, in my group, we have typically three faculty members. So besides myself, the Chancellor Block, and usually I like to have someone doing their sabbatical from another country come and work here. Okay. That sort of rotates from year to year. Like this year, we have a Professor Kim from Seoul, Korea, who's working with us. Next year, it'll be Professor Tahara from Tokyo, Japan. But so I like to have a mix of faculty. And then typically we have a couple of research associates. One, Don Lowe, who's been with me for many years. Okay. Sorry. And then there's typically another couple of postdocs, and then I like having graduate students in the lab. I think they're fun. So usually we have right now we have three graduate students, or four graduate students in the lab right now. And then because I started doing research when I was 18, right? So I was a teenager. I think it's really important to have undergraduates involved in research. Okay. So we typically have usually about 10 UCLA undergraduates working with us at different degrees of honors projects and just different things. Sometimes even high school students in the summer, but not during the year. Okay. So it's a mix of people with different career goals and aspirations, and that we're sort of try to keep together and working together. It's definitely, that's part of the challenge of my job now.
And how do you keep them motivated day in and day out?
Yeah, well, right. It does vary. I have not. I'm fairly flexible about people come and work with me with different career goals and aspirations in mind. I mean, I try to give advice and including motivation depending on what they're trying to do. Some of the people are definitely going a traditional academic route, and that's things that I'm most familiar with. But frankly, a lot of the students, graduate students, have found great success going into more teaching positions after they get their PhD. And there's a lot of good jobs out there, and for some of the students that have come through, I mean, one of my last PhD students within one day, had a faculty position, but it was at a teaching, a place where mostly he's teaching, but the pay is good, and and he likes what he's doing, and so it's so for just to give you so in his case, he did more teaching than some say another student who would be interested in more academic. So I really try to make the training individualized for each of the people. Okay,
that's it. That's good. And what else? I think what are you going to talk about today? And whenever you get a chance, you can present your work.
Sure, great. Yeah. So what I've prepared today is sort of a relatively short presentation, starting off with really like 5 or 6 slides that are sort of distilling what I view as sort of the key things that you need to know. And anyone needs to know about circadian rhythms. Okay, so it's really just very general introduction. And I give my apologies to any of my colleagues that are out there, that may be listening, because this is not really this part of it. I'm just going to be sort of broadly summarizing a lot of work, including work that my friends have done,
Okay.
in the few slides. But then I want to convey one of the key ideas that that we're thinking about in the lab and trying to communicate, this idea that there's really a sort of the healthy sleep-wake cycle is fun, sort of fundamental to our wellness. Okay, and disrupted rhythms are just something which has so many impacts on our health broadly. And then what does that mean then in terms of possible treatment options for patients that may be particularly vulnerable to these disruptions? And I'm going to share a little bit of data with you. That's some of our newer research, where we're using these principles of what I'm going to call circadian medicine to actually treat a mouse model of Huntington's disease. Okay. And so that's sort of what I had planned to go through today, assuming I can get the PowerPoint working on this on the Google Hangout. Yeah. So that'll be a challenge here. You'll have to bear with me. So we'll see how this works. So I haven't done this before.
Here you go.
Is it working?
Yeah.
Okay, good. So then I'll just go through this presentation, and again, it's titled Circadian Medicine because I'm really going to be trying to emphasize the links between circadian rhythms and health. And so just by way of just again very broad introduction, what the heck is a circadian rhythm? This is something that comes up all the time. And I think one of the key things that we all have to remember is that this is a endogenous rhythm. So it's a rhythm which is generated from inside our bodies. This is something, although, of course, humans, we've all known about sleep-wake cycle and rhythms in behavior, but the assumption had been until relatively recently that this was due to the external world and not due to a process inside us generating it. So this is one of the key concepts. Is that it's endogenous rhythm, that the cycle length is close to but not equal to 24 hours. That's where it gets this name, circadian. So a cycle length which is about a day, about 24 hours. In order to perform its function as a biological timing system, it has to be synchronized to the environment, and light is the predominant cue which synchronizes these clocks. Although there's other things as well, and we've become very interested in the role of the timing of when we eat as another synchronizing cue that has a lot of impact in humans. This timing system impacts about everything in our body. And here in this slide, I'm just showing some different examples. I don't know if you can see the cursor or not, but these are just some rhythms and core body temperature. But of course, any behavior, blood pressure, the secretion of hormones. Here I have melatonin, which is high at night and low during the day. Cortisol, the stress hormone, which is high before we wake up in the morning and starts to rise. So basically, almost every hormone, almost every behavior, physiological process, even the turning on and off of genes within the cells in our body are regulated by this clock, this timing system. And so given this widespread influence, you should not be surprised. This is a system which is under strong genetic control. It's a genetically determined system. This is a cartoon version, and this slide I always feel particularly guilty about because most of the field of circadian rhythms is focused on the molecular genetic underpinnings of these oscillations. So there's literally thousands and thousands of papers on this and hundreds of really good labs working on this, and I basically try to summarize it in a cartoon. Okay, so if you'll just bear with me, this is a cartoon showing this is the nucleus, this is the cytoplasm, so this is just a prototypical cell, and basically at the beginning of the cycle, so you can imagine dawn, the clock and BMAL bind a specific site in the DNA known as an E-box, and drive transcription of a number of genes, but including the 3 period genes that we have in mammals and 2 cryptochrome genes. Okay, the genes will be activated. The mRNA will get translated into proteins out in the cytoplasm. The proteins will accumulate over time. They dimerize and accumulate. And about 12 hours after the beginning of this cycle, they translocate back into the nucleus, where they will, through mechanisms that are still a little bit uncertain, but basically they will bind the clock and BMAL and move it away from the E-box, turning off the transcription. Now, there's many elements of this which I'm leaving out in this oversimplification, but this key concept of a negative feedback loop, which is present in most cells, maybe all cells in our body, is one of the key concepts in this field. Okay, and what are the genes that are regulated by this molecular feedback loop? Well, a lot of different genes that maybe 8 to 12% of the genome. So depending on which tissue you're talking about, it's a different set of genes that are being regulated. So for example, in the nervous system, where I mostly work, it'll be genes involved in synaptic transmission and metabolism. But in the liver, it'll be a whole different set of genes, or the heart, it'll be a different set of genes. So we have different genes that are regulated by this timing mechanism. So okay, so that's another one of the key concepts. So one of the things that has come out of research in this area, which I think is super cool, is basically what I think is the discovery of a new sensory system in humans, because the process of working to see how light was activating and regulating these molecular clockwork led to the discovery of a novel photopigment known as melanopsin by Iggy Provencio and some other scientists. So this is a picture here of the retinal ganglion cell layer stained with an antibody against melanopsin, and I think you can see that. So there's the cell bodies and then these processes that go out. So basically, it's like a plexus that's in our retina. And these cells are directly light sensitive, okay, due to the presence of melanopsin. Now, they also take in light input from the rods and cones, but they are directly light sensitive. And the DNA structure actually most resembles an invertebrate photopigment. So I like to think of it that we have a bit of an invertebrate or a bit of fly, actually in our eye, which is detecting the light signals. And is particularly sensitive to the blue and the green wavelength light, which has a lot of implications for our health. As a lot of us are, and we'll come back to this maybe later in the talk, but just to sort of foreshadow it right now, many of us are living in environments that during the day we're under fluorescent lights, which have a lot down. The wavelengths are mostly in the reds and yellows. And then at night we come home and we're on the computer or video game, and that has a lot more in the blue-green wavelength. And this timing system will essentially follow the light, so that for many people they'll start having an inverted light-dark cycle essentially, by not being outside during the day under artificial lights and exposing themselves to the computer monitors at night. This will drive, in and of itself, this will drive sleep and circadian dysfunction. So, okay, so let me, okay, so this light sensing system, which is completely separate from the visual cascade, by the way. I mean, this does not go to the vision at all. So you can be completely blind in the visual sense and have a perfectly normal light input to the circadian system. And this shows then this picture here is of a rodent suprachiasmatic nucleus, or SCN, which is the central clock in all mammals. The light input here would be going into these red cells, and then we have these other cell populations up here. But here's sort of there's a cartoon version with this melanopsin sensitive, blue-green sensitive retinal ganglion cells would then be synapsing onto a subset of cells within the SCN circuit. these cells have to synchronize the rest of the cell population, and they do this using the neurotransmitter GABA, as well as some neuropeptides, especially vasoactive intestinal peptide, VIP. And together, these two cell populations then drive the outputs. They synapse onto a number of output pathways, including, probably most importantly, the autonomic nervous system. So the whole balance between sympathetic and parasympathetic system varies as a function of the time of day. Also, all the major arousal centers in the brain, like, for example, the raphe that releases serotonin, or the substantia nigra releasing dopamine, or, locus coeruleus, any of these modulatory structures are receiving input from the SCN. So literally the hormonal environment, the neuromodulatory environment in our brains varies as a function of the time of day. There's also, as I mentioned before, the SCN synapses onto cells that are controlling the secretion of hormones. So that's why we have the rhythms and melatonin and cortisol. So this is basically a system that has a very simple input, okay, the very simple input being this light input here, going to a very defined nucleus, the suprachiasmatic nucleus, which is the central clock, but then the outputs spread all over the body. So the outputs are very diverse, okay, to communicate with the rest of the body. I should mention, because one of the things, and don't worry, I'll be finishing up on the basic stuff soon, but one of the things which is really critical to the function of the SCN, and one thing that we've devoted a lot of our own attention to, is that these cells are spontaneous pacemakers, in the sense that if you take the cells and you record from them, so here's an SCN neuron with patch electrode, that the cells themselves will go and have spontaneous oscillations in neural activity. So all day long they're firing action potentials, and during the night they're electrically silent. And that's critical for them to communicate this information to the rest of the body. So this is sort of then the discovery that most of the cell population, certainly all the major organ systems in the body, also have their own molecular clockwork. That we sort of have a network, okay, of oscillators that control our circadian behavior in sleep. So, and this is just a schematic showing that, where we have the light input going into the central clock in the SCN, the central clock communicating via the hypothalamic pituitary adrenal axis, as well as the autonomic nervous system, to synchronize then clocks, independent timing system, which are found in things like the heart and the lungs and the liver, the islets within the pancreas. Okay, so we have a distributed network of clocks within our body. Okay, and the reason why this matters, okay, this matters, and I'm just about done with the introduction, but the reason why this matters for so many disease processes is what we're finding is that the signals from the central clock become compromised, and each of these different organ systems start doing their own things. They lack the central timing mechanism. And this just sort of highlights, again, I didn't want to go through all of these different models, but basically in a variety of mouse models of both aging and disease processes, what we're consistently finding is that the SCN neural activity itself, okay, the output from the clock, is what's being compromised. The molecular clockwork seems, in all these different models, seems to be working fine, at least early in the disease process. So it's a question of communication, okay, a question of signals having difficulty going from the central clock and reaching all these outputs found throughout the body. And what does that mean for us? Okay, so a variety, I mean, many studies, not just ones done by my group by any means, but broadly speaking as a field, what we've found is when you disrupt the circadian output, you get a set of symptoms in whether it's people or whether it's in our mouse models or flies, for that matter. Okay, so you get cognitive disruption, especially memory problems. Okay, so you can generally learn but not remember what you've learned. okay, when you have a disrupted circadian timing system. For those that are genetically susceptible, we also see evidence for triggering affective disorders, especially depression. Very, very rapidly, we see disruptions in the metabolic system, and this basically, even young healthy people start developing the endocrine profile of a type 2 diabetic within about 5 days of having a disrupted circadian cycle. Okay, so it's very, very rapid. GI disturbances, which occur rapidly over time. Luckily, not soon, but over time, cardiovascular disease, and even increased risk for certain cancers. And here the data is strongest in women for breast cancer. Mostly epidemiology at this point, but with a biology that makes sense because the circadian system regulates cell cycle. So these are the sort of symptoms, and in fact, most of the audience has experienced these before, because you'll get a taste of this just with jet travel. Jet lag will give you, many of these same symptoms. Luckily, that's just a transient disruption of the sleep-wake cycle of the circadian system, and you'll recover within a few days. Mm-hmm. But imagine being chronically jet lagged, right? Always being in that state. And we believe many of the patients here, indeed, are in that state of chronic jet lag. And so this is sort of the idea, then, sort of part of the key idea that I really wanted to leave with you today, because it's one that we're really focusing on, is, so if we just envision the disease process as a combination of genetic mutations plus your environmental perturbations, and most diseases seem to have both of these components. Okay, originally when we started the work, well, I started thinking about, okay, so the disease then was leading to the sleep and circadian disruption. Okay, and certainly it's the case that, pretty much as I mentioned earlier, every patient that we see in here has a disturbed sleep-wake cycle. Okay, so that's leaving you to think that, okay, the sleep and circadian disruption is a very sensitive but not very specific marker of ill health. Okay, but the thing that I want to, that we're focusing on right now is I've illustrated here by this red arrow. Okay, in the previous slide, I just mentioned, well, if you have a disrupted sleep-wake cycle, you have all these other symptoms, right? And basically, you have patients with all the neurodegenerative diseases, with the neurodevelopmental diseases like autism. The patients aren't sleeping for decades. Okay, and so we think that actually may be triggering a lot of the additional pathology within these disease process. And why that matters, that's just a hypothesis, but why that matters is it raises the possibility that if we went in and if we could treat the sleep and circadian dysfunction, that we could actually alter the progression of diseases caused by something else. Okay, and that's sort of the key idea that we're working on right now. Can we treat incurable diseases by stabilizing the patient's sleep-wake cycle? Okay, so that's the key idea that we're trying to do. So what are the things? What are the things that we can do? Okay, just practical things. These would be helpful for anybody. Okay. So one is be in a robust photic environment. Okay, it turns out that it seems that most people, at least in the developed world, are only spending like 5% of their time outside. Okay, and so the lighting makes a big difference. The lighting of where you are. If you're going to be inside, the indoor lighting is huge. And basically, you want to expose yourself to the blue-green wavelength lights during the day, and you want to avoid that at night, if while you're sleeping. Okay. Likewise, another thing is scheduling meals. Okay, that late night snacking will disrupt your sleep-wake cycle, and also add to your waistline too. So try to avoid it. Right. So and exercise, of course, exercise is generally beneficial, but the timing of when you exercise also matters. That if you're trying to influence the sleep-wake cycle, having regular exercise during the day and avoiding it at night is definitely what you want to do. Now, if you have to stay up late, okay, then late night exercise can be perfect. So these are sort of lifestyle things, right? That will influence the circadian cycle and will improve most people's sleep-wake cycle. But what we're arguing is that the patients with with a variety of diseases may be particularly vulnerable. Okay, that we all probably can benefit from these sort of lifestyle change. If you're dealing with a patient that has a disease, that they may be more vulnerable to these disruptions, and certainly in the mouse models we're finding evidence for that. Now, of course, the general logic in the medical community is that patients won't actually change the way they live. So there's a lot of interest as well in developing new pharmacological tools to alter the sleep-wake cycle. But that's not our research. Our research is really focusing on these lifestyle things. And if you're interested, I'll just mention that I've just recently edited a book that's on this topic that is just available now. So if you wanted to delve into it in more detail. Yeah. Okay, so basically in the last part of the talk, I just wanted to show some experimental data. Okay, I've mostly been talking very broadly, and I wanted to share some of our data that we the last couple years we've been working on a study where we're looking at a mouse model of Huntington's disease. And just to remind the listeners, Huntington's disease is an incurable genetic disease. Okay, we know the gene. If you have the mutation in the Huntington gene, CAG repeat, depending on the size of that expansion, you absolutely will get the disease. And furthermore, the length of the expansion is directly correlated with when you're going to get the disease. So a longer expansion means you get the disease earlier and more severely. Okay, that being said, we believe, and we have evidence now in the mouse, that we can actually alter the course, the progression of this disease by doing these manipulations that I just talked about, these things that are simple, cheap lifestyle changes. Okay, and I'm just going to show you a little bit of data from mice, and to give you a little bit of background here, the way that we're doing these experiments is that the mice are in a cage where they have access to a running wheel. Okay, and they can choose to run on the wheel or not. It's this voluntary activity. We don't force them to do anything. Mice normally love to run on the wheel, and in fact, they run for kilometers every night if given this opportunity. They're all like little long distance runners, as it turns out. And we just monitor the number of revolutions of the wheel, and then we've been working with we've been working with several models, but I'm going to show you data from this model where this is called the BAC HD model. So using the BAC technology, the bacterial artificial chromosome technology, basically the human gene, the human mutation is placed inside the mouse. Okay, and this shows their early in the disease, before they have motor symptoms. This shows their wheel running activity, and you can see they're showing, so this is 1 day, 2 days, 3 days. The data is double plotted, and then successive days are one on top of each other. And they're nocturnal animals, so they're showing most of their activity during the night. And this is then the same line of mice, if you look to the right, under what's called a line feeding, where they're under a schedule in which they can eat as much as they want, but only for 6 hours. So it's a 6-hour feeding, 18-hour fast cycle every day. And so that feed fast cycle has been shown to have some benefits in and of itself. But in terms of looking at the circadian system, that we can improve the activity rest cycle. And especially, I think one of the things that you can probably appreciate here is during the light, when the mice should be sleeping, there's actually a lot of activity in the animals under ad lib conditions. And that's one of the things which is really dramatically improved just by controlling when the mouse is eating. Okay, so the scheduled feeding seems to improves the activity cycle. It also improves sleep behavior. So this is monitored. So here we're monitoring sleep with a video system, semi-automated video system, where we're just following the mouse's behavior. So it's not using the EEG recordings, although we do sometimes do that. But in this case, for screening purposes, we like to use the just this model of monitoring sleep behavior, and we and others have shown it's about 95% correlated with the sleep measurements that we do with the EEG recording. But anyway, the behavior, so basically during the animal's sleep, the schedule of feeding, this feed fast cycle, is improving their sleep, and actually also the activity. So both sleep and activity are improved by this manipulation. But okay, so I expected to improve the sleep-wake cycle by controlling the timing of eating, and we've also done this with light exposure, for example, as well. But what about the key deficit in this genetic disorder, which is in the movement? You know, the patients are characterized by, well, originally by dance-like movements, uncontrolled movements, and what we're finding is that the scheduled feeding, although it does not restore these animals in any way to wild-type levels, but does improve their motor performance. Okay, compared to the ones that are on the feeding schedule, have better motor performance. And this is just some data that here, where they basically, the mice they're removed from their home cage and placed in a foreign cage on one side, and essentially they have to walk the plank. It's like a ladder placed on its side, and they have to walk the plank from one side to the other to get back to their home cage. And we measure the number of errors that they make, and you know, under these conditions. And what we're finding is that we can both decrease the number of errors and also improve the time that it takes them to cross, just by putting them on this feeding schedule. So here's some examples of the errors so at 6 months of age, so the number of errors in the free feeding mice, just the ad lib fed mice, are shown in the white bars, and in the dark bars here is the number of errors when they're on this feeding schedule, okay, itself. So their motor performance by this. Here's another behavioral test where they're basically placed on, the mice are on this wheel that starts slowly rotating, and it's called a rotorod, and basically it starts accelerating and gets faster and faster, and you just basically monitor how long it takes for the mice to stay on the wheel. And again, so here's the data from the free-fed mice, and here's the data from those on this feeding schedule, and so they're basically staying on the wheel, you know, more than twice as much, okay, which is a nice indication of their improved motor performance, as well. So basically, we're treating the mice, and this is just some you know, one example, but we're treating the core symptoms of Huntington's disease just by controlling the timing of eating and improving the sleep-wake cycle. And so we're very excited about this, and of course exploring the mechanisms underlying it. And that's what a lot of our work is involved with right now, is both Huntington's disease, but then also some mouse models of autism, because of course the autistic kids also have a very disturbed sleep-wake cycle. And so we're working to try to figure out first in the mouse models what can be done to improve the sleep-wake, but then also what is the impact on cognition and social behavior in these mouse models. Okay, so just a little, that was all for the data, and just sort of to summarize then, you know, what I'm, you know, shared with you today. Okay, the circadian timing system regulates the temporal patterning of most processes in our body, and you know, data from this field, it clearly indicates that disruptions in the circadian timing system has wide-ranging impacts on our health and well-being. Okay, disease and aging are disruptors of the circadian timing system, but you know, critically, it's also how we're living our life, right? It's our modern lifestyle of exposing ourselves to light, going and getting, you know, the midnight meal. You know, these are things which are disrupting, okay, the normal temporal patterns in our body, okay? And now what can you do about it? Well, the defenses against this disruption, okay, whether it's caused by disease or you know, how we're living, involves living in a temporally structured environment, paying attention to lighting conditions, the timing of meals and exercise, and again, there are a number of new approaches, pharmacological manipulations that the companies are trying, and we've evaluating some of those, but that's certainly not our focus. And with that, I'm happy to have any questions.
Okay. So can you get out of the stop sharing, Chris?
Oh, yeah.
It's a pretty nice presentation. So I enjoyed it. I have a few questions. One of them,
Sure.
have you looked into any gene expression, or like you are not looking into that part of it? How does it go?
No, we always look at gene expression. I was trying to keep it very
Okay.
very sort of straightforward, okay, for this presentation. So for all of our studies, we always have the behavioral component that I shared with you today,
Yeah.
but we also have a lot of physiology, both electrophysiology at the level of the central clock, but also measuring rhythms in cardiovascular function and core body temperature. We also look at the anatomy of how that's altered of the timing system, but obviously critically for any of our work is how does this alter the molecular clockwork, okay, because that's really at the source of how these rhythms are generated. Now what we found in general, okay, and it does vary with which disease, but in many of these disease models we're finding the molecular clockwork is still working, but what's happening is its ability to influence the rest of the body is being reduced. We see this with aging, and we also see this with many of the other diseases. So we're not finding many examples, although there are some, but we're not finding many examples of where the disease is directly altering the core molecular clockwork itself. There's a lot of, even if you go in and try to mutate the core clock mechanisms, there's a lot of compensation that occurs, and it's hard to get this is the system which is so essential for our health that there's a lot of protection built into it. Okay, but in the disease process, what we're finding is communication. Now, the good news about that is that means some of these external environmental scheduling type things can actually improve that communication. And in the case, for example, of Huntington's disease, we just recently looked at 100 of the genes that were most impacted by the disease process, okay, that have been identified in other studies. So does this sort of feeding schedule alter the genes whose dysregulation is thought to be core to the disease? And in 20 out of the 100 genes that we looked at, we found the scheduled feeding improved the gene expression. Okay, so not every gene. Okay, we didn't rescue everything. We didn't restore these animals to wild type animals, but actually 20%. If you want to look at it that way, of the genetic markers for this disease, were actually the transcription levels were improved by these manipulations.
Okay, and like there is a lot of talk these days about the mitochondria, where you have the energy, amino acids. And the food intake happens. I mean, like it's a big source for all the energy. So have you looked into any of these mitochondrial aspects or anything? Did you see any genes getting affected?
Yeah, some of the genes that are altered. Well, okay, so first I should back up and say that there's a variety of evidence, not from my group, but from other groups. Okay, the circadian system also alters, the basic functions of the mitochondria. So that's already known. It is also known that especially the neurodegenerative diseases alter mitochondrial function. Okay, so it makes sense that there's a linkage, but we have not been able. That's something we haven't yet done. Is to ask whether these environmental manipulations that we have been improving the behavior and physiology of the animals, are they altering the mitochondria? But something like scheduled feeding, I mean, that seems very reasonable. That would also alter, especially the process of autophagy. Like if I was betting right now, that's where I would bet that we would see changes, but we don't have the data to address that.
Okay. And these different neurological disorders, like you seem to say that there is some connection with the sleep pattern and all those things. Do you see any specific signatures for, like, for example, Huntington disease? You have certain signature which is different from other diseases, anything like that?
Yeah, I mean, that's a great question. And at this point, I we put a lot of effort into trying to find something that would be very specific, like a specific biomarker, as it were.
Yeah.
You know, what we see in the mouse model is a fragmentation of the sleep-wake cycle, and so basically the animals don't stay asleep and they don't stay awake during during their active cycle.
Mm-hmm.
There's also a loss of the precision. Normally they would start their day at a very precise time, and there's also a tendency for a phase delay, so they tend to be starting their day later, and beginning it, starting, so a delayed phenotype. And when we look in the Huntington's patients, so we now have a Huntington's clinic here, and so we measure with the patients as well, and we see a very similar phenotype, okay, in the patients, as we see in the mice. Okay, so there's a lot of similarities in terms of the nature of the disruption. All those symptoms that we saw in mice, we also see in the patients. But that's different. That's not exactly an answer to your question. So what we don't know is, we don't have, like, I can't say, okay, that sleep disruption would only be found in a Huntington's patient. Okay, that's what we don't have. So right now, the way I think about it is that these sleep and circadian disruptions are that something is, something's wrong. Okay, but don't give you a specific, indication of what it is. Okay. Which disease is it? Because there is an example in Parkinson's. So the Parkinson's patients have something called REM sleep disorder, at least a subset of them, I should say. And if you have REM sleep disorder, so that means during REM sleep, your motor systems aren't appropriately shut down. Okay. So basically, the patient would start acting out their dreams. They would move around, thrash around a lot. So that's highly correlated with ultimately the people getting coming down with Parkinson's later on. And so that would be a case where there was a specific marker. But in the case of Huntington's, we haven't found that marker yet.
Okay. Yeah. I think it's pretty interesting, and I think there is lots needs to be done, and I look forward to more of your work in future. And thank you so much for taking your time and giving us a nice presentation.
Absolutely. Really, yeah, no, I'm glad to be here, and for your listeners and viewers, I guess, who would be interested and have specific questions, please feel free to email me.
Thank you so much, Chris.
Thank you. Okay, bye.